Good morning, my dear students. Today I will discuss about the germination process in case of maize and bean seeds. So, in case of bean seed, it is showing apical germination and the maize is showing the hypogeal germination. So, in this case of the bean seed, you see what you will have. It is showing apical germination. Then you have the cotyledons, the food is stored in the cotyledon, and when seeds are sown, it is absorbing the water and radical comes out by the elongation of hypocotyle, which carries the cotyledons upward. Simply means in case of epigeal germination in the bean seeds, you will not be observing the cotyledons. Okay, means in case of germination, the cotyle, hypocotyle, it remains inside the soil. It remains inside the soil, and you will not be able to see the cotyledons or the radical plumule first. When it will burst open, then after some time you will get to see this type of arrangement. But in case of maize monocot seed, what you will observe? You will see that the hypocotyle comes above the soil, but in epigeal germination it is below the soil, it is under the soil. Okay, this is the only difference. Now if you will talk about the different parts, the cotyledonary leaves, they will be formed, the two cotyledonary leaves will be formed, that's why it is called dicot plant. When in case of maize, only one cotyledonary leaf will be formed, that's why it is called monocot seed. Okay, so if you will talk about the maize, that is the monocot endospermic seed. So the section will consist of many parts. The first will be seed coat. So already you are knowing about the seed coat. The seed coat means it is protecting the inner parts of the seed. Next is the endosperm. Endosperm is the part which is just storing the food. The stored food material is in endosperm. Okay. Then the separation, if you will talk, the epithelium layer, it is separating the upper embryo, lower embryo and the endosperm. Okay. The special layer is present, that is aileron layer. Okay, which contains only proteins. The endosperm contains stored food, mostly in case of in the form of starch. Okay. Then the embryo, if you will talk, the embryo, it is the lower part of the seed. Then there is a massive cotyledon on the embryonic axis, that is called scutellum. The embryo has two main parts, radical plumule. Now the radical, they are covered by coleorhiza and the plumule is covered by coleoptile. These are the coleorhiza and coleoptile protects the radical and plumule respectively. Okay, so in case of germination in maize monocot seed, so if after absorbing the water, the grain produces radical and forms the root system. Okay, this is the only thing, the difference. So if you are comparing, we are here only comparing the maize grain and the bean grains. The comparison is first the monocot and dicot, and the next is epigeal and hypogeal germination. So it is showing, maize is showing the hypogeal germination, where you will see the leaves up above the soil. But in case of bean seed, it remains inside the soil. Rest everything is same. Okay. Now embryo, hypogeal and epigeal germination. So when the coleoptile is above the soil, the plumule bursts out and it forms the leaves. So you can see the leaves. Okay. The maize grain, it is showing the hypogeal germination. It remains under the ground due to the increase in epicotyle region size. After germination, the young seedlings drops into saline water with the radical end downward. Radical will be downward, plumule will be upward. Okay, just like the same. Maize plant also, there also radical forms the root, plumule forms the shoot. Okay, so this is the germination. Rest you can study from the book. Very easy. It is very easy. Only you have to remember some parts, what it is called, the definitions. So these are important. Rest everything is same. Now coming on to some experimentation related to the germination of seeds. So there is first experiment where you have to show that water is necessary for seed germination. So what you have to do, you have to take two petri plates A and B, and you have to place blotting paper in both of them. Then you have to moist them with putting some water into it. Now place some bean seed or gram seed on the wool or blotting paper and in the dry cotton of the petri plate B. So what you have to do, in first plate you have to take some water, in the second plate you don't have to take any water, it should be dry. Okay, place both of these at room temperature 25 to 30 degrees for a couple of days. So what you will observe, seeds in petri dish A where you have put some water will germinate, but in the number B it will not germinate. Okay, and so it concludes that water is necessary for seed germination. So another is there, suitable temperature is responsible for seed germination. Then there also you have to take same thing, two petri plates you have to take A and B, then place some green or gram or bean seeds on wet cotton wool in both of the petri plates. So here in this case you have to take both of the petri plates and you have to wet both the petri plates. Keep the petri dish at one place, one at the one place and other at low temperature, means one at suitable temperature 25 to 30 degrees and other at 2 to 3 degrees. So what you will observe after examining, you will find that the seeds in A germinate, while seeds in the refrigerator where you have put the temperature 2 to 3 degrees, it will not germinate. So this shows that a suitable temperature is also necessary for seed germination. Okay, third is experiment, you have to show that oxygen is necessary for seed germination. So two conical flasks you have to take and then you have to place some wet cotton wool in each of one and put the bean seed or gram seed, whatever you know, whatever you are having. Now you have to hang these two test tube containing water in flask A, and there what you have to do, if you are taking these two petri plates, okay, so in one of the petri plates, suppose this is A and this is B, you have to put here pyrogallic acid in number B. So what the pyrogallic acid is doing, the pyrogallic acid absorbs all the oxygen. Okay, and here you have put the seeds. Here you have put the seeds, and here you have moist cotton, and also you have moist cotton. So what you will see, seeds in flask A will germinate, while seeds in flask B will not germinate. The reason is flask B is containing the pyrogallic acid. The pyrogallic acid absorbs all the oxygen, and here so oxygen is not present, and due to the deficit of oxygen in the flask B, germination will not take place. Okay, this is a simple experiment which proves that oxygen is necessary for germination. Last experiment is given that water temperature and oxygen is essential for germination. So what you have to do, you have to take one flask and there you have to take one wooden plate and you have to put some seeds. 3 seeds you have to put. One should be completely immersed in soil, means water. So this is your A, B and C. So what you have to do, in seed number A, it will be totally immersed in water. Seed number B, half immersed in water and half it will be above the water surface. And in number C, it is totally above the water surface. So afterwards, you will have to put this arrangement in the sunlight, and what you will see that A number will have poor germination, B number will germinate quickly, easily it will germinate, and C number also it will not germinate. The reason is water, temperature and oxygen, they all are necessary. It is getting temperature and oxygen, but it is not getting water. C number, that's why it will not germinate. B number is getting water, temperature, oxygen, everything it is getting, so it is germinating. But in number A, what you are observing, that it will not germinate since it is not getting oxygen, it is only getting water and temperature. So this experiment proves that all three things are necessary for the germination of seeds. Okay, so you revise the topic, this chapter, and if you will have any problem, we will discuss in the next class. Okay, thank you.